Assalamu alaikum. My name is Saqib Shahbaz and I am a student of Scientific Research, Medical and Social Institute, Jalalabad. Today I am going to explain my topic is the complement system. So the complement system is a part of our immune system that enhances the ability of the antibodies and the phagocytic cells. So the complement system is also known as the complement cascade, which means that it continues till the precursors of the complement system is also known as the complement cascade, which means it continues till the precursors are available. It is a part of our innate immune system, so it remains same as in the living organism. It consists of many small proteins that are synthesized by the liver. So there are 30 different proteins and protein fragments that are present in the complement system. So there are three different pathways, which is the classical pathway, the alternative, and the lectin pathway. So we are going to explain the classical pathway. The classical pathway is considered to be the part of the specific immune system because it relies on the antibodies that initiate it. So the classical pathway is triggered by the C1 complex. The C1 complex stands for which contains the C1q, R2 and S2, which consists of one molecule of C1q, two molecules of C1R and two molecules of C1S. So the activation of the C1 complex occurs when the C1q binds to the IgM or IgG antibodies complex with the antigens, or it can also be C1q binds when directed to the surface of the pathogens. So here is a diagram which shows when the antigens are present on the microbe and C1 complex binds, it further converts into C2 and C4, which further divides into C2A, C2B, C4A and C4B. Here the C2A and C4B combines to form a complex which is named as the C3 activation complex. So C3 activation complex, as we know, was formed by the C2 and by the binding of C2A and C4B. So it is formed on the surface. So the function of the C3 activation complex is to form and activate the C3 proteins. This is done by cleaving the C3 into further two parts, C3A and C3B. So C3B, as we know, was divided by C3 activation complex. So these molecules are produced by C3 activation complex and these bind to the surface of the bacteria. And C3A increases the inflammatory response by binding to the mast cells and help them to release the histamine. So next is the C5 activation complex. C5 activation complex is formed by the combining of C2A and C4B, which was making the C3 activation complex. So it has a slight affinity for binding with the C3B. So when C3B binds to C2A and C4B, it forms the new complex which we named as C5 activation complex. So C5 activation complex activates and it further cleaves into C5A and C5B. So many C5B activation proteins are formed which begin to coat the surface of the bacteria. So it forms membrane attack complex, which is known as which we say as CMAC, which is represented in capital letters. So C5B on the surface of the bacteria binds further to the C6, which binds and activates it, and so it can C6 can further bind to the C7, and it forms a chain which further binds to C8, C9, and it forms a long chain. So these proteins form a circular complex. This is named as MAC, which is membrane attack complex. So this causes the cytolysis in the cell. It forms the circular membrane as it forms a channel in the cytoplasm in the cell from which the cytoplasm rushes out and the water rushes in. So So here the inner integrity of the bacterium is compromised and the bacterium is died. So here is a diagram showing the cytolysis where the loss of the cellular contacts is going through the transmembrane channel and it forms the membrane attack complex. So this is a flow chart that shows the membrane attack complex in front, which here we have the C1 complex. It further divides into further branches. So the C2 and C4, C2A and C4B further binds and form a complex, and in the next step C3B and and these binds and form the C5 activation complex and the chain continues.